Good morning, ladies and gentlemen. Good morning, Adjarn. Sawasdee ka tukon. My name is Kyla Christian Eve Sanchez, a fourth-year international student from the Philippines studying my bachelor's degree in food science and technology under the Faculty of Agricultural Technology. And for today, I would like to present to each and every one of you fermentation properties of yeast isolated from cacao fermentation at Nangua Community Cooperative. I would like to start off with the introduction. So as we all know, Thai cacao has been produced mostly in the northern part of Thailand, and it has given potential income for smallholder farmers. The Thailand cacao market is expected to grow annually by 6%, and it is one of the tropical agricultural products par excellence, coffee beans. Next, we have the problem. So the natural fermentation of cacao using banana leaves to retain the heat created during the process, and it may give advantageous yeast, but as well as microbial agents to the cacao fruit. So it can produce uncontrollable natural fermentation. It gives the presence of biological contamination, and it may reduce the efficiency of the fermentation process, and it may also be able to give us less productivity or less produce if we focus on the natural fermentation process. Now, when it comes to the objective, this research focused on isolating yeast from natural cacao fermentation and to identify the different properties of the yeast that has been isolated, and as well as to investigate the enzyme activities that are relevant to the cacao fermentation of the isolated yeast. The purpose of this research is to report on several properties of yeast that were isolated from the natural fermentation of cacao from Nangua Community Cooperative, and the study scope is restricted to a small-scale experiment laboratory. When it comes to the research concept, yeast ferment the glucose of the cacao pulp into ethanol, which can perform pectinolysis and produce flavor compounds onto the cacao to achieve a good quality of natural cacao. So here are the yeast fermented. It can create glucose to the cacao pulp into ethanol, and with the ethanol, we can perform pectinolysis, produce flavor compounds, and of course achieve a good quality of natural cacao fermentation. Now, what are the expected benefits? So yeast starter culture initiates the development of the flavor precursors that are necessary for the organoleptic quality of the cacao to better control cacao fermentation. So to know the different properties of the yeast, to select appropriate strains for starter cultures and develop a good way for consistent cacao fermentation. When it comes to the literature review, we have the importance of cacao as commodity. So according to a study about Thailand's homegrown craft chocolate culture conducted by Mr. Damekau in the year 2020, in Thailand, cocoa has actually been growing for over a century, and over the last few years, this fruit has been produced in the southern and northern part of Thailand, and it has been shown potential income once again for the smallholder farmers. And as well as based on a research conducted by Statistics on the year 2020, the revenue in the cacao segment in Thailand amounts to 720 million in 2023, and its market is expected to grow annually by 6%. Cacao fermentation. According to a study conducted in the year 2021 by Guzman and Ramos about fermentation of cacao, it was stated that in order to obtain fine cacao beans, it is necessary to carry out the process of fermentation. So why? It adds interesting occurrence of the microbial activity. It is a key process for the formation of the characteristic of the chocolate flavor, and it is the first step in the chocolate making chain that can make the organisms grow for 5 to 7 days of the process. We have the roles in the cacao fermentation. So, according to a study of the functional roles of yeast by Diaz Munoz and DeVos in the year 2022, yeast initially perform an alcoholic fermentation, consuming the pulp carbohydrates to produce ethanol and carbon dioxide, and displays pectinolytic activity to degrade the cocoa pulp pectin. So number one, it is essential. The yeast is essential to flavor development. It is also essential to the final acidity of the cacao beans. It produces ethanol and it displays pectinolytic activity to degrade the cocoa pulp pectin. What are the microorganisms involved in cocoa fermentation? So, a study by Hamili and Susiwala on the year 2018 stated that fermentation of cocoa beans involved the role of microorganisms in outlining of polyphenolic compounds, proteins, and sugar. So we have here the microorganisms involved in the fermentation of cacao. We have yeast, different types of yeast, LAB or lactic acid bacteria, AAB acetic acid bacteria, and other miscellaneous such as molds and bacteria itself. Now, what are the importance of using starter culture in cacao fermentation? So, the whole cocoa fermentation process can be better controlled if we use starter cultures. It gives more uniform quality of the cured cocoa beans. It gives faster fermentation, improve the quality of the roasted and cured beans, and it improves the quality and the sensory profiles of the end products. We have here the yeast starter for cacao fermentations influence the flavor characteristic of the cocoa beans, and of course, it improves the quality and the organoleptic properties of the cured cocoa beans. So when it comes to the enzyme production, according to a study conducted about the functional yeast starter cultures for cocoa fermentation, cocoa pulp is a rich medium for microbial growth with 1 to 1.5 pectin. And because of this, the pectinolytic enzymes are the key to the good quality of fermented and dry beans. Here are some relevant researches about the said research. We have here, according to a research conducted by Dia Munoz and Yvonne, strains of Saccharomyces cerevisiae and Pichia kuryavzevi are usually well adapted to stressing condition of cocoa fermentation, and these yeast are best performing at highest temperature, and it is the biggest driving force behind the die-off yeast upon the research fermentation process. And as well as on that side, the Saccharomyces cerevisiae, Pichia cholera, and other type of yeast species that are used as starter cultures for cocoa fermentation of the research that has been conducted on the year 2017 by Gosneto and Sokol. Now let's proceed to the materials and methods. So for the sample collection, materials used were the isolates from the natural fermentation process of the cacao originating from Nangua Community Cooperative in Mung Phetchabun District, Phetchabun, Thailand. The samples were taken from two different areas of fermentation. We have near banana leaves and, of course, by the cacao pulps itself. So the sample were serially diluted 3 times and we have used the spread plate method onto the DRBC agar. On this left side is the near leaves sample, and on the right side we have the isolates that was taken from the cacao pulps itself. So it is diluted 3 times. We have dilution 1, 2, and 3 over there. Next, we have the materials and equipment. We have used different types of agar, yeast nitrogen base, and another type of equipment such as analytical balance, petri dishes, cap bottles, microscope, and methylene blue. And of course, for the identification of the yeast, we have used the API 20C AUX identification strips. Now, when it comes to the preparation for isolation of yeast in Nangua Community Cooperative, we have taken samples from 2 different areas near the banana leaves and in the cacao itself. It has been spread plate using the DRBC agar from different locations in the fermentation box. It has been incubated for 3 consecutive days at 25 degrees Celsius. Now for the agar preparation for the selection of the colony, we have used the PDA agar. We have transferred it into the blue cap bottle with the 400 ml distilled water and autoclaved for 15 minutes and transferred to the 20 petri dishes. We have used PDA agar for this research. And next, when it comes to the yeast isolation based on colony characteristic, we have analyzed the different characteristic of the colonies present in the DRBC plates that was isolated from Nangua. And for this one, we have actually streaked a very small portion of the colonies onto the PDA agar and labeled it according to characteristics. For example, we have the flat surface, the round surface, and different other characteristics that we have seen from this DRBC plate. And we have collected about 30 to 40 plates in this part over here. And we have selected this thick colony and examined it under microscope. Now, for the morphology under microscope, we have dropped 1 methylene blue on the slide and add a single colony for each plate under the microscope slide. We have examined it with a microscope, and of course we have with the type of cells and the characteristics of the cells that was shown in the microscope, we have selected suitable colonies for further examination or analysis. So the various traits of the colonies that were examined more thoroughly are given particular names beginning with PCB, which stands for Petribune. So yeast isolation based on colony characteristic, once the suitable colonies has been identified, we have seen the sizes and characteristics of the cell through a microscope. We will be streaking it at a malt extract agar plate and restrict it until we got the pure cultures, or we have obtained a pure culture so that we can use it for further examination, and we store it in the fridge after we have got or obtained the pure culture. Now, for the yeast identification using API 20CAUX, 5 ml of distilled water was distributed into the honeycomb wells, and a portion of the yeast colony was picked up using a pipette and was placed inside the test tube with NaCl 0.85 NaCl. Now, an ampule of the API medium was opened, and 10 ml of the previous suspension was transferred into it. Now, the lid was placed on a tray at 29 degrees Celsius for 48 to 72 hours for the identification of the yeast. Now, for the interpretation of the strip, on the results sheet, you will be able to see 4 groups with numbers 1, 2, 3, and 4 indicated for each of this one. And by adding the corresponding positive reactions within a group, a 7-digit was obtained. And with the use of the API web identification software, we would be able to read and understand what kind of yeast that is, or identify the species of yeast that was presented in using the API strips over here. For example, for this one, we have the PCB142. It has shown that it is a species of cholera. Now, properties relevant to cacao fermentation. So the pectinase enzyme production, the culture media was prepared with YNB, or yeast nitrogen base, with 10% agar and 1% pectin. All of the media components were autoclaved for 15 minutes at 121 degrees Celsius, and we have inoculated the yeast colonies onto the media. For this one, we have PCB 140, 141, 143, and 142. And if the yeast can hydrolyze pectin, a clear zone would appear around the colonies. For the fermentation experiment, the sterile liquid medium was prepared with yeast nitrogen base, 15% glucose, and 1% pectin. The liquid medium was incubated at 30 degrees Celsius for 5 days, and samples were taken every 24 hours for pH and TSS analysis. Now we will proceed to the results and discussions. For the selection of colony, the colony characteristics are shown in Table 1 for straight plates from the isolated plates. So we have three different isolated plates. We have cocoa 1, 2, 3, near leaves 1, near leaves 2. And these four different columns over here shows the different characteristics of the plate. So we have flat surface, round surface, pink dots, and dark pink dots. The yeast colonies that were selected were classified as streaked in this table, whereas the colonies that were streaked were labeled as not streaked. Now for table 2, here are the selected colonies for further analysis that has been named PCB for Pechabun. We started from PCB 135 ending to PCB 143. So before I would like to focus on this one, we also used this type of table to identify it through microscope. Here we have PCB 135A and up until letter H, which is PCB 143. So based on the morphology of the cell under microscope, the yeast were chosen to proceed for further investigation. That is why in this table we have showed that from cocoa 1 we have 5 different isolates that was chosen for further examination. For cocoa 2, none, because it hasn't shown any type of characteristic that stood up. And for cocoa 3, we only focus on the transparent characteristic. Near leaves 1, we have a white web kind of characteristic. For the PCB 136, it came from near leaves 2 or day 2 of the DRBC plate, which is from the characteristic that has a round surface. And lastly, for PCB 141, it came from the near leaves plate of the DRBC agar from the flat surface characteristic. Now over here, this is the morphology under the microscope. So for PCB 135, 136, 137, and 135, 136, and 137, you can see that it has a ovoid shaped budding. So you can clearly see it from the morphology under the microscope that it has the possibility to be a Candida species of yeast. Now with the morphology under microscope, for example, we have the PCB 142 that is letter G. You can see that it has a ellipsoidal or like a little bit of pointed shape. So it might give us a clue that it might be a Colecara apiculata type of yeast. So with this part of the research, we will be able to somehow have a little glimpse of what type of yeast are we doing for further, or we chose for further examination. Now for the yeast identification using API, we have seen here at table 3, here are the positive and negative results for the different active ingredients for each yeast. So here are the active reagents for each isolates. As you can see in this part over here, we have numbers and what type of reagents or active ingredients are in this different isolates. So for example, we have number 1, it has a D-glucose reagent. Number 2, it has a glycerol reagent. Let's give PCB 138 as an example. For the 01, it is the control, so that we would be able to see if a turbid part of the well of the API strip has been, there's a difference between the control. So we have here for number 1, it has a D-glucose reagent. For number 2, it has a glycerol reagent. But for number 3, PCB 138 is negative when it comes to calcium to ketoglutamate type of reagent for the active ingredients of each isolates. Now for the identification, we have here table 4. It is the yeast identification from the API TM identification page. So we have concluded that the yeast isolates 135, the identification obtained with a numerical profile shows that it is a Candida famata. PCB 136 is Candida guillermondi. PCB 137 is the same species of yeast. 138 is a Cryptococcus homicola. PCB 140 is Candida gilmorti. PCB 141 is Candida famata, and PCB 142 is Cloacara apiculata. PCB 143 is Candida lusitani. Now for the properties relevant to cacao fermentation, for the pectinase enzyme production, this table shows that each of the yeast does not produce pectin for the pectin, is negative for the pectinase enzyme production with the YNB or yeast nitrogen base with the 1% pectin in the agar. And then we have here the pectinase enzyme production. Here we have figure 4.2. It demonstrates letter A, the absence of clear zone, because the clear zone for formation by yeast will show the positive result. But for the yeast that has been isolated from nangua, it shows a absence of the clear zone formation by each of the yeast that has been present. And for letter B, this is the positive control using a pectinase enzyme drop on the agar. And letter C is the negative control using another type of yeast, which is Saccharomyces cerevisiae. So as you can see, A and letter C are somehow the same. So it means that the samples does not give or does not have a clear zone formation by yeast. So it cannot break down pectin. For the fermentation experiment, for the pH value, we can see over here that the different isolates has a distinct type of pH value. During the course of the Collinell's fast 5-day incubation with each isolate added and labeled, results revealed that the pH values of the 8 varieties of yeast isolates changes. So as you can see, the PCB 135 variety experienced the greatest pH increase, which has been from 2.8 to 3.25, and it has so the pH values of the majority of the isolates have declined on day 1 in the same manner. However, PCB 142 has produced distinct results, increasing on day 1 and then sharply falling on day 2. And the greatest pH values for all isolates were obtained on the 5th day. Now we will proceed to the TSS value. Results revealed that the TSS values of most isolates shows a non-moving results from day 0 to day 2, but decreased dramatically on the following days. The yeast isolates from the PCB 143 variety experienced the greatest TSS decrease from 20 bricks of day 0 to 13 bricks of day 5. And on the 5th day, the lowest TSS values were attained on all isolates. For the conclusion and recommendation, so we can conclude that 8 yeast isolated were successfully evaluated through the API-TM identification software, where we identify 1 Cryptococcus homicola, 1 Chlora apiculata, 1 Candida lusitane, and 2 Candida famata, and 3 Candida guillemondi. Results have shown the absence of clear zone formation on plates, but therefore bacterial species should be utilized to produce pectinase and break down pectin polysaccharide compounds. The pH value of the 8 different yeast isolates changed during the course of the 5-day incubation period. PCB 135 has the largest pH shift from 2.8 to 3.25. And for the TSS values of the 8 variety, the isolates share the same pattern of results from day 0 to day 5, but PCB 143 showed the same TSS value from day 0 until day 2, but decreased dramatically on the following days. So the results have shown promises that this yeast could be potential used as starter cultures for cacao fermentation. For the recommendation, examination of each yeast strain for the development of the cacao flavor should be carried out, and screening of more yeast and chocolate products. Thank you.